Welcome to Cancer Lesson 5.5, How Will Cancer Be Treated in the 21st Century? The goals of this lesson are to explore why driver mutations are crucial to the development of new cancer treatments, and to identify factors that limit our ability to identify specific mutations that cause different cancers. At the end of the lesson, the students should be able to explain the importance of driver mutations in cancer progression, explain how personalized medicine designs drugs to target driver mutations, explain how genetic sequencing is used to identify somatic and inherited driver mutations, and explain the concepts of prevalence and penetrance of mutations. We will achieve our goals by developing a personalized cancer treatment plan based on data from the Broad Institute. To prepare for this lesson, you'll need to review the following key scientific concepts that will be presented throughout the lesson. Each cancer has its own genetic fingerprint. For example, two people who have lung cancer most likely do not have the same mutations. Cancers are increasingly classified based on their genetic mutations rather than which organ they originate in. Personalized treatments aim to target the driver mutations in cancer cells responsible for abnormal proliferation. Driver mutations drive the cell to become cancerous, whereas passenger mutations are mutations found in cancer cells that did not cause the cell to become cancerous. These are simply along for the ride. You can review this scientific content in the background reading provided for you in the teacher primer, the teacher manual, and the student workbook. The teacher primer provides in-depth knowledge about the scientific content presented in this lesson. The teacher manual or lesson plan provides a minute-by-minute explanation of the lesson structure, and the student workbook provides additional explanation for students. Be sure to print the lesson handout, the lesson worksheet, and the homework worksheet. The key points of the Do Now are that cancer is a disease caused by DNA mutations, and identifying which DNA mutations are present in the tumor is key to targeted treatment. Some mutations that are present in the tumor are only passenger mutations, meaning they have no effect on cell proliferation or cancer. Other mutations in the tumor are driver mutations that play an active role in transforming the cell. We start the Do Now by asking, what mutations do cells need to transform? If we look at a cancer cell, we find that most mutations don't have an effect on cell transformation. The ones that do have an effect are called driver mutations, and harking back to lesson 3.5, there are three types of driver mutations. The first type affects tumor suppressors, leading to increased survival. The second type affects telomerase and promotes anti-aging. And the third type affects proto-oncogenes, which leads to proliferation. So how do we distinguish driver mutations from passenger mutations? The green mutation is clearly not a driver mutation, because when it occurs, the cell dies. The blue mutation is a driver mutation, because it leads to the cell dividing into many more cells in just one generation. The orange mutation could be a driver mutation, but we cannot tell because we don't know what happens in the next generation. The pink mutation looks like a passenger mutation, but it could be a driver mutation if it affects cell survival or anti-aging. Next, have the students figure out which of these tumors will grow the fastest, and discuss what possible treatments could be used against them. For example, students may come up with a treatment plan that involves only targeting cells that have the blue mutation. Would treatment change if we knew that these tumors belong to different organs? These days, cancer treatment aims to treat specific cells with certain mutations, rather than the entire organ. This is a key point of the lesson. Every person's cancer is different, and yet most of the time, given limited treatment options, we have to treat them as though they're all the same. One way to personalize medicine is to divide the population based on what similar tumor characteristics or driver mutations their cancers have. By segmenting the population or dividing the population and administering treatment based on what driver mutations their cancers have, we can more closely reach an optimal treatment response. In the activity, students will work in groups of 3 to complete an interrupted case study on designing personalized treatment plans based on evidence about the efficacy of different chemotherapy drugs. You will need the lesson worksheet and the lesson handout, which details how well different chemotherapy treatments kill the cancer cells in each sample as a heat map. Dark red means least effective or least cytotoxic, and the dark green is the most cytotoxic. Read the introduction in the lesson worksheet for more detailed instructions. Next, we introduce the differences between somatic and germline sequencing. Somatic sequencing identifies mutations present in the tumor, whereas germline sequencing identifies what mutations are present in every cell. Using germline sequencing, we can ask if any of the mutations found are known to be associated with cancer. This leads us to 2 important terms that help us determine risk: prevalence and penetrance. The higher the penetrance of the mutation, the higher the risk of cancer if you have the mutation. But prevalence of the mutation gives us an idea of how likely you are to have the mutation in the first place. Note that the key points of the activity are that driver mutations, not the primary organ, are most important when designing tumor treatment plans. This is why tumors from different cancers, such as lung and brain, may sometimes respond more similarly than tumors from the same cancer, such as lung and lung. The wrap-up leads us into a discussion about what might happen if everyone's genomes could be mapped and mutations could be tracked over time to determine what types of treatments could be developed. Companies and databases such as the Cancer Genome Atlas have sequencing information that includes all the gene mutations in many types of cancers. Students should realize by now that it's very important to find the driver mutations that lead to cancer. But finding mutations alone isn't enough. We also need to know if that gene is active or on, or silenced or off. This field is called epigenetics and is an active area of research. For homework, students should read an article on the Cancer Genome Atlas and start preparing for their final discussion. One of the most common questions students ask is how has cancer treatment changed with recent research about each cancer's molecular characteristics? As we understand that cancers have different molecular characteristics, we have begun to treat tumors rather than the organ in which the tumor resides. Another question students ask is, can you find driver mutations simply by sequencing DNA? Not necessarily. Even if we can sequence every cell in the body at any time, we still won't know what genes are silenced or active. This is called epigenetics. The epigenetics can even vary from cell to cell. At the end of the lesson, collect the lesson worksheets to assess students' understandings of what the 3 types of driver genes are and how to identify them. This lesson demonstrates how knowledge about driver mutations and other genetic factors that predispose cells to become cancerous is important in how we treat cancers. Don't forget, if you have any questions, concerns, or feedback, to let us know. You can contact any of the CTSE team members and we'll be happy to help you.